So I've been involved in Parkinson's disease for about 16 years, and my primary motivation for focusing all our efforts on Parkinson's is to identify novel neuroprotective strategies and therapeutics that might slow or halt the disease process in people affected by Parkinson's disease. There are no currently accepted neuroprotection therapeutics available for Parkinson's that would be expected to slow the disease process. And bringing to the clinic one of these for the first time takes hundreds of groups and a huge amount of resources. Ultimately, we don't know what causes Parkinson's disease. We don't know all the specifics about how it progresses. But we think LRRK2 will give us a window into some of the insights we need to not only target LRRK2 as a neuroprotection for Parkinson's disease, but potentially other targets that we identify along the way. And so with all complex diseases, there's complex responses and there's heterogeneity. And so one of our big focuses in the lab is using biomarkers to tell us who could best benefit from a LRRK2-directed therapeutic, in addition to the number of people with Parkinson's that show kind of this abnormal LRRK2 phenotype that tells us something about the makeup of Parkinson's disease that we don't already know. In all biofluids, in humans, but other species as well, we have a type of microvesicle that we call exosomes, and they're basically shed products from cells where you can capture them in biofluids using very specialized techniques and then survey proteins like LRRK2 that you wouldn't otherwise normally be able to without a tissue biopsy. And since in Parkinson's disease the brain is affected and we don't normally biopsy the brain, we had to find a second source to survey this protein, and it turns out that this exosome source is powerful in that regard. The proteins linked to Parkinson's disease are all relatively difficult to detect, but this new fraction in biofluids that's really just been utilized in the last few years in biomarker research seems to be positively impacting people's ability to detect these protein targets. And so, in the near future, we're going to identify the first neuroprotective therapy that'll begin to slow the disease process. And I think that once that first neuroprotective therapeutic is developed, the others won't be too far down the line. And I think what we've seen in kind of success stories for complex diseases, that you have to approach the disease from multiple angles in an informed way, and people call these precision medicine approaches, where not the exact same treatment regimen is going to produce the same results in everybody. What we are thinking is that there are patient populations in Parkinson's disease that would benefit from LRRK2 inhibition, for example, better than others. And what we've learned, particularly in cancer research, is that biomarkers are going to be critical not only for selection for particular neurotherapeutic, but also for active feedback as to how the therapeutic is responding in disease. And so there's a number of biomarkers that I think will play into kind of successful not only identification but use of a neuroprotective strategy. And so biomarkers will allow you to pick the people who you think would benefit best from a novel therapeutic.